Welcome to MSB Talks, a podcast about science for everyone. Our guest today is Dr. Geoffrey Greene, who is currently the Virginia and D.K. Ludwig Professor in the Ben May Department for Cancer Research at the University of Chicago. His lab studies hormone receptors, including estrogen, androgen, progesterone, and glucocorticoid receptors, using structural biology and PDX models to develop novel therapeutics for breast and prostate cancer. This episode, we talk about Dr. Greene's journey through his scientific background, research interests, and thoughts on the future of hormone receptor targeted cancer therapy. This is MSB Talks. We're glad to have you. And now for our conversation with Dr. Greene.
I mean, we're some Michigan students, but I appreciate you helping us out regardless and talking to us about our project and about what you do at U Chicago as well.
Okay.
Um, yeah, I guess just to start, kind of maybe a little like background about your research and kind of what you did to get to where you are today.
Sure. Well, it's a long time. I've been doing this for 50 years. So, but I got a PhD in synthetic organic chemistry at Northwestern back in 1974, I guess. Um, and then I felt like synthetic organic chemistry had kind of reached its peak. I was wrong about that, but nevertheless, I wanted to get into something that I thought was more uh, biologically relevant. So I did a postdoc at the University of Chicago working on estrogen receptor and breast cancer because the group here, headed by Elwood Jensen, that was his field, and I heard him give a talk at Northwestern. So anyhow, that's how I became a postdoc. And then I never left. I could have left many times, but
Mhm.
I was offered a faculty position after a few years. And so I stayed on, and I've continued to work in the same area for that entire time, which is interesting. I never would have predicted that. But so my research has focused on understanding the biology of hormone-dependent breast cancer, ways to treat it, ways to prevent it, um,
Mhm.
ways to stop metastasis. And I have made use of my organic chemistry. I synthesized an estradiol affinity column to purify the estrogen receptor when I joined the lab here, and that allowed me to
Mhm.
generate antibodies against the receptor, and later on to clone it and do all the things that we've done since then. But it really started with all of that.
That was awesome to
Yeah.
hear.
Yeah.
Um, I guess just going a little bit more into depth on your research, is there any relatively recent finding that you found interesting?
Sure. Um, so I become definitely more translational as time has gone on. I feel like what I'm working on should benefit people.
Mhm.
And so mechanistic work comes out of that, of course, but really focused on trying to better understand and develop compounds that target the estrogen receptor and are useful in prevention and treatment. And the latest one that we've really focused on is a compound called lasofoxifene. It's a selective estrogen receptor modulator. It's been around for a long time, but it was never and it was approved in Europe along with a number of other compounds in that same class to prevent osteoporosis. Um, and it turns out that most of those drugs, or all those drugs, actually reduce the risk of breast cancer in women who were getting them for osteoporosis. But the only one that really got developed into a therapeutic drug was tamoxifen, and lasofoxifene, I think, was probably ignored because it has blended characteristics. So it's estrogenic in some tissues and anti-estrogenic in the breast. But since there was already tamoxifen, nobody pursued that. But then a small company based in Columbus, Ohio, called Sermonix, contacted me several years ago and said they had gotten a license to lasofoxifene, which had been given up by Pfizer, and was I interested in helping them out? So we were doing animal studies. So we work with an animal model, a mouse model, um, in which we inject breast cancer cells directly into the ducts through the nipple. And this is based on some work by an investigator in Switzerland, Catherine Briskin, who showed that when you do that, you have a much more natural environment for breast cancer cells. So what most people do is they inject cells directly into the mammary fat pad. And what happens is that luminal breast cancers, which is the most common, quickly become basal-like. And so it doesn't really reflect the patient that they came from. And so this approach is more realistic in terms of having a model that's more appropriate as a model for human breast cancer. So we've been doing that for several years. And we've gotten, with our help, this drug lasofoxifene has been moved to clinical trials. It's actually in a phase 3 clinical trial now for treating. Oh, and I should mention though that another thing we discovered along the way was that in women who were treated with aromatase inhibitors to block estrogen production, that these tumors that are ER positive develop mutations in the estrogen receptor that become activating, and they don't need hormone anymore. They also become more difficult to treat because the affinity for drugs that target the estrogen receptor is reduced anywhere from 10 to 20 to 30 fold. And so they're no longer in a viable clinical dose range. So lasofoxifene turned out to work effectively on these breast cancers that are metastatic, and they have a mutation, one or more mutations in the estrogen receptor, and they're particularly effective in those patients. And so a small clinical trial, a couple of them, showed good efficacy, and they were given to women. Lasofoxifene was given to women who had already progressed on hormone endocrine therapy, like tamoxifen and fulvestrant, and they had to have a mutation in the estrogen receptor that was activating, and that represents about 40% of all ER-positive breast cancers. So it's a very significant portion of the breast cancer population. So anyhow, in the small trial, it showed that lasofoxifene really extended the duration of response beyond, even though they'd already had endocrine therapy. And now there's a phase 3 trial to test that out. And in the meantime, several other companies have pursued a similar route, but using SERDs. And I know you asked that question already of me. I haven't answered it yet, but lasofoxifene is a SERM. It's a selective estrogen receptor modulator, so it behaves differently in different tissues. SERDs are really designed to degrade the estrogen receptor, which if you think about it logically, it's like, oh, that would be great. We just want to kill the estrogen receptor, and that should be a more effective drug. And that's still the thinking of some major pharmaceutical companies like Lilly and Genentech, a couple of others. And that's the route they're pursuing. And the reason I don't like that is because, and this relates to another question you asked, which is the bystander effects elsewhere in the body are problematic because estrogen receptor is expressed in many tissues, and you don't want to kill it in tissues where it's important. You only want to really kill it in tumor cells or in cells that directly affect the tumor. So lasofoxifene is good in that way because it's more estrogenic, even though it's very effective in women with advanced breast cancer. So it has, I think, better properties and can be used earlier. So the ultimate goal, from my viewpoint, is prevention. But prior to that, you want to be able to use a drug in as wide a population as possible so that you get benefit all the way around, quality of life as well as effective therapy. So that's been one of my big focuses recently.
Mhm. Yeah, I mean, that sounds really exciting. Would you say this is kind of like the culmination of all your work? Like you've spent, you said 50 years doing all of this. Would you say this is like the thing that you're most excited about, or what other kind of
Yes, at this point,
frontiers?
I would say so. I'm also just recently taking a look at GLP-1 agonists.
Okay.
There's reasonable evidence that they seem to delay progression of breast cancer in women who take them.
Mhm.
So we're exploring a different aspect of that, which is whether or not they might be usable in combination with endocrine therapy. So that if our model bears that out, and we don't know yet, we're in the middle of it, then I would want to expand that area because that would literally affect millions of women. So many people take GLP-1
Yeah.
agonists now. So but in general, what I'm working on really is ways to reduce mortality and risk of hormone-dependent breast cancer. I don't focus really on triple-negative breast cancer, although that's very important. It's just a smaller, it's more difficult in a smaller subset of women. But along the way, we've cloned the estrogen receptor gene. We've uh, solved structures of dozens of compounds bound to the estrogen receptor to try to figure out how they work, why are they more or less of an agonist or an antagonist, and can we exploit that property? How do we avoid having deleterious side effects outside of our target, which I know you're interested in also. Um, so yeah.
Cool. Yeah, you brought up this idea of like antagonists and agonists and kind
Mhm.
of classifying the two. Um, in the broader field of cancer biology and drug discovery, is there anything that kind of excites you in that regard, like new discoveries in drug discovery?
Yes, I have a clinical fellow in my lab who's working on a couple of antibody drug conjugates, and I have to say that that area has exploded pretty quickly. And also, I see that Eli Lilly has an antibody drug conjugate against the receptor you're interested in, and it's in clinical trials. Um, so the advantage of those drugs, if they're targeted correctly, is to make them as tumor cell specific as possible
Mhm.
and minimal side effects. Now, it turns out the most well-known one is trastuzumab deruxtecan, and that targets the HER2 receptor, which is present in about 20% of breast cancers, whether they're hormone dependent or not. Um, but HER2 is expressed elsewhere in the body. Uh, and so there are some negative side effects of those drugs, but it turns out that their effectiveness seems to significantly outweigh the side effects like neuropathies, nausea, that type of thing. And now there's some others. And so now the latest, there's no doubt that the latest idea now is that any membrane receptor exposed protein that is cancer selective, no matter what the cancer is, somebody's going to develop an ADC against that. That is definitely what's going on right now. So did I didn't hit my hand.
No, I think when you raised your hand, it
Ah,
registered.
Yeah.
oh, that's AI.
Yeah.
Yeah.
Yeah. There you go. That's the
Yeah.
Anyhow, so yeah, I think that's a really big area. It goes along with a related area, which is a little bit different, but does sort of the same thing, are protacs and molecular glues, small molecules that target proteins for proteasomal degradation. But of course, the trick there, as you want to know also, is you want it to do it in your target, your tumor cell. You don't want it to do that elsewhere. And that's always a potential problem. And certainly all of the receptor-based drugs that are out there are not 100% tumor specific. They target other tissues as well, which is an issue. Now I know
yeah.
you have a peptide, I guess, or you're working on a peptide against the receptor. Um, I don't know whether you've thought about trying to conjugate it to a degrading drug.
Yeah, our approach is like we kind of thought about this recently, but you can essentially frame our project as an ADC, but without the linker.
Okay.
Um, because in a sense, what we're doing is we're using like a magnetotactic bacteria,
Mhm.
right? And then we're taking this surface peptide, just expressing it constitutively,
Yeah.
such that it can bind folate receptor alpha. And then upon binding, we induce a second plasmid, which has like a heat shock promoter, and then
Yep.
that will produce like a cytotoxic payload.
Okay.
So that kind of two-step delivery is what we're thinking about. Does that sound like anything meaningfully different than an ADC?
Oh yeah, I think mechanistically it's definitely different. You get a similar outcome, uh,
Hmm.
but the way you're going about it is a different pathway. So yeah, and I did see that you were doing something with bacteria. So how are you getting them to? So then accessibility is the only thing I can think of offhand.
Hmm.
Not, the advantage of antibody drug conjugates, for example, is that they will go everywhere in the body for sure, and they won't be impeded for the most part getting to tumor. Although tumors have their own problems because they have their own barriers to things coming into them. That's actually, just as an aside, that's one of the advantages of the kind of drugs that I work on, which these small molecule drugs basically they penetrate everywhere. There isn't any cell they can't get into, but that's both good and bad. But in your case, what? Yeah, tell me a little bit more about accessibility to tumors. How specific is it to? Are they to tumor cells?
Yeah, so I think starting with folate receptor alpha, that I think is overexpressed in 70 to 90% of high-grade serous ovarian cancers. Um, and I think obviously there is a meaningful distinction between like solid and liquid tumors. Like CAR T cell therapy obviously faces the same barrier, right? And the sense that cells are a lot harder to navigate than, small molecules. Um, but yeah, essentially all of our specificity kind of lies, well, there's like kind of a three-pronged approach. One is that the bacteria themselves are aerotactic in the sense that they seek out low oxygen environments, the tumor microenvironment, which
Right.
would be one of them. A second part of them is that they're magnetotactic by nature. So they respond to external magnetic fields, meaning that if you were to take an external magnet and kind of guide it along the body, you could take a population of cells to the tumor site.
Yeah.
Um, we're
I've
also using like,
that before. I'm a little bit aware of that as a possible approach, but don't know much about how well it works.
yeah. And
Yeah.
then, yeah, we're also using like fluorescent markers, right? We have like a fluorescent tag that will fluoresce when it's in a low oxygen environment.
Mhm.
And then the last little element is that surface peptide, which shows specificity for the folate receptor.
Right. So kind of where do you stand right at the moment? I mean, how far along are things? And
Yeah, so I know Jack's been working on the science side of this a lot, but we are in the process of assembling like a dual plasmid system to kind of get our bacteria into shape. The first plasmid is under a constitutive promoter. It'll express the surface peptide at all times. That's essentially the goal of that. I believe it also houses our fluorescent protein. And then the second plasmid is under a heat shock promoter that encodes a pore-forming toxin called cytolysin A.
Mhm.
And once the bacteria reach a critical temperature, which will induce through an alternating magnetic field, just enabling the dipoles to flip rapidly, causing the bacteria to move rapidly. Um, so once that critical temperature is reached, only then does the payload get released towards the tumors.
Okay, that's good. So
Yeah.
now, did you have any animal data, or this is all still?
No, this is
Or-
all mostly theoretical. I know we've got one of the plasmids fully constructed, and I think we're pretty close to finishing the second plasmid. I'm not sure if it's been done quite yet. Um, but so we're getting the two plasmids constructed, and then we're gonna start get them into the AMB1 bacteria pretty soon. So.
So I assume other people have done something along these lines, not specifically what you're trying to target, but there must be data on bacteria being selectively attracted to low oxygen tumor environments. I don't know not sure what other environments in the body might also be targets, but I guess if they don't express the receptor that you have a secondary target for, then that's the advantage. I like the fact that you have a two-step approach. So you have to satisfy both in order to get effectiveness, but that also should tend to reduce
Intensity, yeah.
other effects. Yeah.
Mhm.
Yeah.
So I like that. That's an advantage over antibody drug conjugate, because those are just one step, basically.
Mhm.
Yeah, and then kind of uh
Sure.
going back towards your own work.
Okay.
You have like a fair bit of experience with obviously targeting ligands towards receptors and kind of getting treatments towards tumor microenvironments.
Yep.
In general, what kind of issues do you see with drugs in the microenvironment? All right, it's obviously a low oxygen environment. Cancer cells are absorbing large amounts of glucose, so it's um, often often has a different nutrient profile than the rest of the body.
Sure.
Essentially, how should we kind of approach that?
So that's a great question, because it's something that we're interested in. I have to say that up until recently, most of the people in my field really haven't considered much about the tumor microenvironment. A lot of hand waving, but not real data. And so there's several aspects of that I think are important. And what we do know, and this is now beginning to show up, is that in addition to the changes in the nutrient environment, as well as physical aspects of the tumor microenvironment, there are cell types in there that express the estrogen receptor. And what we don't know very well, like macrophages for sure, and I think T cells do as well. What we don't know is whether a drug that you're using, let's say it's tamoxifen, so we know that in tumor cells, tamoxifen is an antagonist on the estrogen receptor. We have no idea whether that might be true in the surrounding microenvironments, such as in macrophages or in T cells. There is receptor there, and I'm sure the drug is, interacting with it, but does that help the tumor or does that hurt the tumor? And that's something that we're just beginning to explore, and some other labs are as well. So we are trying to look at that. Part of the limitation for breast cancer and mouse models is that it's very difficult. There are very few mouse models that express hormone-dependent mammary cancers for whatever reason. So that means that but you want that if you want to study the tumor microenvironment, because you need to have an intact immune system. And so there are a couple. We're working with a syngeneic mouse model now that has a STAT1 knockout that was developed by the Schreiber lab in Wash U a while back. And the good thing is, and this is something that I would assume you'll want too, is you want, if you're going to, I don't know if you're going to look at ovarian cancer or breast cancer or both, whatever, but the advantage of that particular model is they do develop hormone-dependent mammary cancers. And in addition, you can put them into wild-type mice. So you don't have to put them back into the STAT1 knockout mice. So you can put them into totally immune competent mice. So that gives us a model now to work with. We've only been working with that model for about the last year or so, but those are the kinds of models that one wants, so that you can really study the tumor microenvironment. And I haven't, my background and expertise is not in metabolism or nutrient environment, but that's a hot area for sure. And what we will learn more about for sure is the immune microenvironment. That's something that we're trying to focus on in this model. But so I can't answer. Well, we don't have information really on what the tumor microenvironment in breast cancer is doing for the most part, because it hasn't been explored. very much mechanistically.
So one big limitation of our project is that we can't use animal models. We're not allowed to, unfortunately, because of the competition that we are in. Um, so our plan, I think, for the time being is just to use cell culture,
Mhm.
right, Jack? I think that's how we're gonna
Yeah.
kind of assess
Mhm.
in
I
vitro.
believe so.
Yeah. Obviously, using an animal study is preferable in terms of like uh, realistic effects than a cell line. But if we are only using a cell line, do you have any tips as to how to kind of optimize our study?
Well, you should try to get to the point where you have something that comes closer to a tumor microenvironment, and you can do that.
Mhm.
You can work with organoids. You can get organoids.
Hmm.
So you don't. You don't. You just have to have a source of them. Which I would assume that you said you're at Michigan State. Is that right?
University of
University of Michigan.
Michigan.
Oh, you're at University of Michigan. Yeah, that's what I thought. But certainly there, the organoids are readily available. You can also get them commercially. But the advantage of that is that it gives you an environment that still, in the beginning at least, represents the patient and the tumor that it came from. So
Mhm.
this is good for solid tumors. So if you're, as I said, like ovarian or breast or prostate, whatever.
Mhm.
Um, and spheroids is another way to go, and you can do that in cell culture for sure.
Mhm.
You can get tumor cells and you put them in the right environment. They will grow as three-dimensional spheres, which again, that makes them a little bit more stem-like, and it gives you properties that are better than just 2D cell culture. The problem with 2D cell culture is you're just selecting for adherent cells on plastic, and you can certainly learn a lot from that, but it's very restricted in terms of how you translate that. So the more three-dimensional you can make your system in a in a representative way, so that it's more like it came from an animal or a human, the better off you
Mhm.
are. You should be able to do those things without having to do any animal research yourself. And I'm sure that, I mean, I know labs at the University of Michigan who do that kind of work. So um, that that's what I would do if I were in your situation. You can also create an environment. So you're doing, I assume, are you doing 2D cell culture? Yes? No?
Yeah,
Yeah.
that's the plan.
Okay. And are you using collagen or matrigel or other environments that will change the properties of your cells? That's something else to consider. Well, you would need to do that if you did organoids anyhow.
Hmm.
So yeah, so there are definitely ways to create a more realistic environment that goes beyond just the 2D environment that you're already working in. And then at some point, you just, if I were you, at least, or what I did, is you collaborate with somebody who
Mhm.
has animal models and you get them involved, and then that would really add a whole new dimension to what you're doing. And you don't have to,
Yeah, for sure.
yeah, you don't have to be doing it, but somebody should be doing it.
Mhm.
I mean, for years and years, well, I did work on animal models way back in the beginning, but a lot of what we did over the years was all based on cell culture, standard 2D cell culture. And, we learned a lot from that, but then eventually needed to move on to working with animal models. And in the beginning, you collaborate with somebody.
Yeah, I mean that sounds yeah, it definitely sounds like a good kind of future direction for the project. I know we have very strict guidelines for what we are and aren't allowed to do for the competition. For iGEM, but definitely after that, getting this research and what we've done and working with with professors at the University of Michigan or wherever it may be, um, seems like kind of the most reliable way for for what we've done to actually go somewhere. Um, I guess one last question about your research and about what you've done and just your experience. Like AI has been a huge thing in in research and academia the last couple of years, and it's been controversial, but also I've seen a lot of a lot of professors, a lot of PIs really taking it up. I know that my lab at University of Michigan that I'm a part of, my PI is very big on AI. So I just would love to hear kind of your take and how you think it might impact both the research that you do and kind of drug discovery as a whole.
Yeah, so we've certainly started taking advantage of AI, at least in a relatively simple aspect, which
Mhm.
is, I mean, it's an incredible search tool. And I've learned that it isn't always right. I definitely it like in this animal model I was just describing to you, ChatGPT tried to convince me that it was uh that it was a like a triple negative breast cancer, and it's like, no, it's not. I don't know where you got that. But um
Yeah.
but I use it to uh actually I've also used it to create research proposals.
Mhm.
And then make sure that you need to validate all the references because it could give you something that's ridiculous. But you can do all of that,
Mhm.
and so it can be it can really be a helpful tool for analyzing your data, of course, for
Mhm.
gathering information out there, for actually um, uh, creating proposals or even
Mhm.
manuscripts. But then, the problem is depending upon where you're getting funding or where you're trying to publish, you have to reveal that. Some places don't like
Mhm.
that. Although
Yeah,
I think it's a mistake to reject it, but on the other hand, people are still afraid of it. So they,
absolutely.
they tend to say, no, you can't do that. At least more and more now, you can, if you just inform people how you used AI and whatever it is that you're doing, and then
Mhm.
then you'll find out whether or not that might be okay or not. But you're not going to stop. You're not going to not use it. Um,
Yeah.
I think that would be ridiculous. So yeah, I think it has great potential actually,
Mhm.
and especially because of the complexity, especially when you get into tumor microenvironment and contributions of the immune system. I think that and immunotherapy, which we haven't even really talked about, but that's another really, I mean, at the end of the game, I think anybody who's doing anything therapeutically now is going to have to have an end goal of engaging the immune environment. That immunotherapy is going to apply to pretty much everything in the future. So right now, breast and prostate cancer are not good targets, although triple negative might be a little bit. Ovarian cancer, I don't think there's any great drugs yet on ovarian cancer that are based on immunotherapy. So solid tumors seem to have lagged behind some on immunotherapy, but that's just a matter of not understanding the system well enough to get them to be targeted
Mhm.
T cells. But it's a complex system. But you won't, you'll be limited though in what you're doing as long as you're just doing cell culture. It'll be, you really need to branch out. I think you need to have a team involved
Mhm.
where people bring in lots of other expertise and models that you might not be allowed to use or don't use, but they do.
Yeah.
And
Yeah, for sure.
that probably my number one piece of advice, actually, is to
Mhm.
spread yourself out some. I mean, remember also, this is something that I think most people, even no matter where you are and what you do, um, don't really get, which is that if you're trying to develop something that you think is going to be therapeutically useful, you just right up front, you have to imagine a 10-year timeline.
Mhm.
The idea that you're going to do something and it's going to end up in people a year from now is definitely not going to happen. And so you're going to be competing with people like, I was just, like I said, with Eli Lilly, they've got this ADC now, and they're testing it in ovarian cancer. So they're going to have long lead time to develop and show whether they have effectiveness or not. And so you're going to be competing against that, but over a period of years where other people are going to have that kind of advantage, other companies. And I mean, no matter what happens in terms of reducing that timeline, which I hope will happen, actually, I think that will happen, but still, it's going to be a long haul. So you have to be somewhat realistic. I review proposals and manuscripts all the time, like, oh, I have this drug and uh, it targets the estrogen receptor a little bit differently than what's out there right now, and we want to develop that. And it's like my first response is that, well, good luck. I believe you. I believe the data you showed me, but your idea that you're going to run ahead with that and compete with what's out there is mistaken. You need to keep that in mind. As I told you before, what I like about your system is that it's different and it's two-step. And that's that I'm not aware that you're competing yet with anybody else in that, or you may be in a lab-based made. I'm not aware you're competing with a company in that, but so that I think that gives you some advantage.
Yeah. I guess one last thing. We ask everyone that comes on the podcast, just if you have one piece of advice for future scientists, anyone kind of high school, college age, what would that be?
Yeah, it's a difficult time right now because it's hard to predict the future. As you know, the current government is not very pro-science, or at least not the way we think about science. And so we get this all the time at the University of Chicago, trying to figure out how to talk to our undergrads and our graduate students about the future. I think you need to look around and try to look forward and see where the issues are. So, for example, as I said, if you're trying to figure out where you want to work, I don't mean a physical location, I mean the area you want to work in. So, for example, I think that the more we know about immunotherapy, the better off everybody's, you're going to be. And so I think that that's not a bad piece of advice to give people, which is look at areas where you can see that 5 or 10 years down the road, there's going to be a demand and a need for that. And then try to go that way. Don't go to some esoteric pathway that sounds interesting and exciting, but that is not going to lead anywhere because you don't always know where something's going to lead. That's also an issue. But so yeah, I would say people should be trying to look ahead as to where things are going. I don't know. We have an organization here actually for our graduate students that exposes them to many other areas actually of investigation where they could use their PhD because the truth is that only a subset of people are going to remain in academia, for example. But again, whenever students come and interview, it's kind of like, what do you want to do? Oh, I'm going to run a lab in an academic institution. And I tell them, I said, well, you have about a 1 in 5 chance of that happening. And so you should be thinking broadly. I think that I think an area that has outlasted almost everything along the way is bioengineering, engineering in general, but bioengineering. So thinking about getting knowledge and expertise in areas that involve engineering is good no matter what you end up doing. So there's no one answer, of course, to any of this, but also to encourage people that, well, first of all, you need to choose something that you're really passionate about, not necessarily something you're good at, but something that you can imagine yourself wanting to work on it 24/7. And if you don't have that, you're probably not going to make it. You'll get further down the road. You can get a degree, probably do a postdoc, whatever, but then you're going to hit a barrier somewhere along the way if this isn't like
Mhm.
your true passion. So I had a chemistry professor when I was in college, and that was his number one piece of advice. He said, find something you're really passionate about and stick with that.
Mhm.
So that would be my advice.
Well, thank you. That's a great piece of advice. I appreciate that. Um, yeah, and thank you for coming on our podcast. We really appreciate you coming out. Yeah, Arnav, do you have anything else?
No, I think that's it. We appreciate you, Dr. Green. Thank you for giving us the
Yeah, thank
time
you
to
so
talk to
much.
you.
Yeah, well, I enjoyed it, and good luck with your project. I think it's
Thank
very
you.
interesting, actually, and it's a niche that not very many people are pursuing. So I think that's in your favor for sure.
Yeah.
So stick with it, but broaden your environment.
Will do. All
All right.
right.
Thank you.
Okay.
Thank you again.
Yeah.
That concludes this episode of MSB Talks, where we delve into the science behind our research into cancer, therapies, and hope for a better solution. We're committed to sharing evidence-based insights and the voices of those advancing our understanding of cancer. To stay up to date with future episodes, subscribe to our YouTube channel, and if you have feedback, questions, or personal stories you'd like to share, we welcome you to reach out, and you can find us on our Instagram at michigan.msbt. Speaking with Dr. Greene were Arnav Sharma and Jack Collier, members of MSBT, and I'm your host, Daniel Yang, and I thank you for joining us today.